So here, we are going to talk about sex linkage, keeping in mind when, we are referring to sex linkage, so we are referring to the genes that are located on the X chromosome. Okay, yeah, because this is going to be described as sex linked, because we already know that male and female, they both will have this X chromosome, right? But female, she will have only X chromosome, while males, they will have X and Y chromosome as well, right? So we will see whether, this X chromosome, which is a sex linked, okay, sex linked gene, whether it is going to carry the dominant allele or not. This is what we are going to understand in this sex linkage. So means we will keep in mind, let me write it here, the gene that are located on the X chromosome. Yeah, we need to keep in mind as I go ahead. Okay, yeah, are described as sex linked. Sex linked. This is very, very important to know. Now, I go ahead with the example. Here, I'll give you the example of what? I'll give you the example of color blindness. Here, let's talk about color blindness. So here, when I'm talking about color blindness, okay, what is that? One of the genes, okay, on X chromosome, it is going to control the ability, right, to see the colors, which colors, red and green color. So in color blindness, when I'm saying, I am saying that one of the genes on X chromosomes controls X chromosome, control the ability to see colors. Which colors I am referring to? I am referring to red, green. Okay, yeah. Now, what is going to happen? Since, these genes, they will be in the receptors known as cones. Okay, where are these cones located? In the retina of the eye. Okay, so now there is an allele of this gene which does not, produce a protein necessary for color vision. So the allele is recessive. So any girl or woman who is heterozygous will have normal color vision. Okay, and males only have one X chromosome. So if they have inherited the allele with recessive, okay, color with recessive allele, means with heterozygous, they will be colorblind. Let's see how it is done. Let me draw it. So here, I am going to draw the phenotype of the parent first. So here, we are going to talk about parental phenotype. So when I'm talking about parental phenotype, you will understand better what I'm saying. Parental phenotype, so here I have normal sighted female. This is normal sighted female, which is going to cross with color blind male. So now what is going to happen? We are going to see here color blind male. So now what is my parental genotype? Parental genotype, when I'm talking about parental genotype, since normal sighted female means she will have both R, capital R. Okay, she's normal sighted. Okay, and male, he will have one X, okay, with the carrier, and will have Y. Now, what will happen to gametes? Now here, what are the gametes which are going to take part in it? So here gametes will be what? Gametes are XR, right? Have I written correctly, right? You guys can see. And here what I'll have one XR, small r, and one Y. Y does not carry this allele since it is sex-linked. Sex-linked means it will be carried by X chromosome only. Okay, yeah, keeping in mind. Now let's go for the Punnett square. So when I go for the Punnett square, here, what will I have? Here I'll have gametes, right? These are my gametes. And here, what will I have? I'll have XR and I'll have Y. These are my two gametes, and here I'll have XR. Now, what is the probability that I'll have? So I'll have XR capital, XR small, and I'll have XR capital and Y, right? Now, what is that? So it means I'll have one female carrier, because as we can see here, right? So this will be what? One female carrier. She is the carrier of that allele. She doesn't have this characteristic for now, but she is the carrier here, as I can see. And this will be what, since this is male, so this one will be normal sighted males. So you see both of them, female, she is going to be the carrier, since, this is the sex-linked disease. So X, which is actually going to carry it. So, and then here, what will happen? Okay, what will happen here? One normal sighted male, because there is no allele which is going to carry this disease. So no carrier for males. Let me write here so it will be clearer what we are talking about. That, the allele is recessive. Let me write here so it will be very clear. The allele is recessive. So when, we say the allele is recessive, so it means if there is heterozygous allele, okay, heterozygous means what? When I'm talking about here, let me put it so like we have all the points very clear. Okay, heterozygous is what? Heterozygous, which is capital R and small r. So here, in this, it will have normal vision when we are talking. Okay, so this allele will has normal vision. Why normal vision? Okay, because here, the allele R, it is, which is actually for color blindness. Okay, so small r, okay, will be for color blindness or color blindness. So unless, males, they carry this small r, they will not be the carrier, or, they will not be the color blind. Understand? Because this is the allele which is actually, for color blindness. Okay. Okay, now, we are going to cross what? We are going to cross both of them. Okay, in the next generation, these two, one female carrier and one normal sighted male. Now let's see how we're gonna do that. So here, let's talk about parental phenotype first. So parental phenotype, here we are going to mention about, I changed the color, right? Okay, so parental phenotype, what do we have? Carrier female, here we will talk about carrier female, and going to cross with what? Going to cross with normal sighted male. Normal sighted male. Now, what is the genotype? So here, genotype, when I'm talking about, let me write it, genotype, I have XR, right, since she is a carrier. And this is normal sighted, so means no small r present, because we know that small r is basically the allele for color blindness. So here, as meiosis occurs, what will happen? We will have one of the allele, this XR, and this is small r. And here what we will have capital R, and we will have what? Y. Now, let's draw our Punnett square. So let me draw the Punnett square. Let's draw it nicely so we can understand. Okay, so here, what I'm going to write? Yes, of course, my gametes. So here, which gamete I'll have? Let's write X capital R and Y. These are my two gametes here. And here I'll have what? I'll have here X capital R and X small r. Right? Now what will I have? I'll have X capital R, as I can see, X capital R. Then here X capital R and Y. Right? Then, here what will I have? X capital R, as I can see, and X small r. And here what will I have? I'll have X small r and I'll have Y. Actually, all of them are different. Okay, so we have to see what is going to be my F1 ratio. Yeah, I have to be very clear when I am writing it. So here, let me write what is my F1 ratio that I get for this. Okay, let me write it. What is that? This is basically one normal sighted female. Normal sighted female, as you can see, both capital R, right? Then, I go down one carrier female, as you can see, one small r and one capital R. Now let's go to male. What will I have? I'll have one normal sighted male. Then next I'll have one color blind. One color blind male. How I have taken all these ratios, I have taken from here. Okay, let me write in front of it so you will understand better what I'm talking about. One normal sighted female, what I have taken, X capital R, X capital R, no carrier in it, right? Okay, then one carrier female, I'm talking about this one. Okay, X capital R and X small r. Since small r is the one which is actually the allele for color blindness, as I have mentioned earlier. Now go to males, as I can see, no small r present. So this is what this is, my normal sighted male. Then here I can see small r present. So this will be what? This will be my color blind male. Okay, so here, yeah, we need to understand that who are the carriers, who are not. Okay, who are actually, so here you see no carrier, right? Because both are capital R. So yes, this is normal sighted. But the second one, even though, okay, she doesn't have color blindness for now, but she carries one allele. Okay, yeah, so this is what we have to understand. Like, let's say if a female is a carrier and a male has a normal color vision, then what will be the chance? 1 out of 4 chance is that one of the children, means 1 out of 4 will have color blindness. Okay, so if, like, let's say if there is a boy, okay, so out of 2, 1 will have color blindness. This is how we understand the sex linkage disease.